So my name is Emily Kim. I'm a PhD student at the University of Guelph, and I'm going to be presenting my project, which is meta-analysis on apparent ileal amino acid digestibility in Eimeria-challenged broilers. So first we'll talk about coccidiosis. So it's an avian parasitic disease caused by the protozoa Eimeria, and its replicative life cycle in the host intestines is what causes significant tissue damage. This of course can lead to reduced digestion and absorption of nutrients, but it can also consequently induce mucogenesis, increase turnover of enterocytes, and activate the immune system, all of which affect endogenous loss of amino acids, thus affecting amino acid digestibility. So why isn't the solution just to include more amino acids into their diet, keeping in mind that we're anticipating some amino acid loss? Well, currently broiler diets are formulated in accordance with the ideal amino acid concept. So if you don't recognize this image, it's called the Liebig barrel, and it illustrates the concept of limiting amino acids. So protein synthesis, represented by the water in this image, is limited by the shortest stave of the barrel, or the first limiting amino acid. So if we were to just supplement more amino acids without taking into account the ratio, the outcome for protein synthesis wouldn't change very much, because the idea is that there exists an ideal ratio of amino acids for optimal growth. So too little of an essential amino acid and protein synthesis is limited. But too much of an amino acid means that it's just going to be excreted as waste, because the body only uses what it needs. However, this ratio can change throughout a broiler's lifespan, so this could be related to age. It could also be related to physiological state. So if we're thinking about a sick bird versus a healthy bird, do they have the same requirement? Probably not, because protein synthesis for growth isn't prioritized metabolically if the bird is sick. But then this also begs the question of whether certain amino acids become limiting during an enteric disturbance like coccidiosis, because different amino acids have different functional roles in the body. So why is this important? Well, as we're continuing to move towards a more antibiotic-free feeding program, and we're limiting what kind of drugs we can use to treat against coccidiosis, the birds may ultimately have to rely heavily on their own immune system to fight off clinical and subclinical disease. So this means that amino acids that were originally allocated for growth and other metabolic processes are now redirected towards fueling the immune system. So having a deeper understanding on what amino acids are most affected by Eimeria may be what we need to identify potential limiting amino acids in hopes of minimizing the detriments of an Eimeria challenge. So our objective of this study was to use a meta-analysis approach to evaluate and quantify the effect of an Eimeria challenge on apparent ileal digestibility of amino acids in broiler chickens. So what is a meta-analysis? A meta-analysis is a statistical analysis that combines the results of multiple scientific studies. So in order to do so, we had to first create our database of publications. So when we were doing our article search, the criteria that we used for our paper selection was, one, that they had to include an Eimeria challenge with at least one control and one challenged group. Two was that they were using broilers. The third was that it had to contain the nutritional composition of the diet. And the fourth was that it had to report measurements of ileal amino acid digestibility. Now, due to the limited amount of studies that actually fit this criteria, we decided to go with studies that reported apparent ileal digestibility instead of including standardized ileal digestibility. So this gave us a total of 13 experiments with 26 treatments. Additionally, we wanted to also see if there was a dose effect on digestibility, so we made a sub-database just for the studies that used Eimeria-challenged, which gave us the total of 7 experiments and 14 treatments. So once we finished our database, we used the program Minitab to look at potential variables and interactions. This could be looking at the effect of challenge, challenge type, challenge duration, age of inoculation, etc. and the significant variables were then retained in the models. We also made preliminary scatter plots like we see here, just to give us a visual representation of the relationship between the dependent and independent variables, as well as recognize any noticeable outliers. All the studies were then verified for crude protein content, apparent metabolizable energy, and SID lysine requirement in accordance with the nutrient specification guidelines of the corresponding breed, just to avoid any confounding effects on amino acid digestibility. So if you recall, we had two databases, the overall database, which contained all the studies. So regression models were fitted with the mixed procedure, looking at the fixed effects of challenge, challenge type, and their interaction. We also ran a 2-sample t-test using this database to compare the difference between amino acid digestibility slopes to create a hierarchy of which amino acid was most affected to least by challenge. The second database was the Eimeria database. So regression models were fitted with the mixed procedure to look at a linear or quadratic response to dose. And for both databases, we used an alpha level of 0.05 to test for significance. So these are regression equations for the impact of challenge, challenge type, and their interactions on apparent ileal digestibility. For the challenge effect, it was significant for almost all amino acids except for tryptophan, such that digestibility decreased with challenge. This makes sense as endogenous loss increases, digestion and absorption are affected, and therefore digestibility should decrease. It was interesting to see that the challenge didn't affect tryptophan digestibility, particularly because acute phase proteins contain a high concentration of tryptophan. So during an inflammatory response like Eimeria, you would expect to see a higher requirement for tryptophan. But if we take a look at the challenge type effect, so this is measuring an Eimeria challenge model compared to a mixed species challenge model, we do see that it was significant for most indispensable amino acids except for isoleucine and leucine, such that the digestibility values of the mixed Eimeria species were lower than that of the Eimeria. So maybe for tryptophan digestibility, it was the combined effect of multiple species that was enough to evoke a response on tryptophan digestibility. As for the interaction effect, it wasn't significant for most of the amino acids except for the branched chain amino acids, where digestibility significantly decreased with Eimeria-Eimeria challenge models compared to the mixed challenge. However, this could possibly be attributed to the functional role of branched chain amino acids and their role in the immune system, in combination with the fact that Eimeria commonly affects the duodenum and jejunum, which are major sites for amino acid digestion and absorption. So here we have our regression equations looking at the impact of dose effect or oocyst concentration on apparent ileal digestibility. We see that oocyst concentration linearly decreased for all amino acids except for tryptophan. We also see that oocyst concentration quadratically decreased digestibility for most amino acids except for arginine, methionine, tryptophan, cysteine, glutamic acid, and proline. So for most amino acids, the effective challenge did increase with the increase in oocyst concentration. So if we recall, we did a 2-sample t-test on our challenge model slopes just to test the difference between amino acid digestibility so that we could create a hierarchy of which amino acid was most affected to least. So we have this ranking here, and we see that cysteine is most affected by challenge, followed by arginine, methionine, alanine, tyrosine, and threonine. So in order to see why, I looked into the literature and found that both cysteine and tyrosine-rich proteins have both been identified in the inner and outer layers of the oocyst wall. Cysteine and methionine are also considered endogenous antioxidants due to their ability to defend against oxidative stress. Alanine, of course, is a significant energy substrate for immune cells like leukocytes. Arginine is used as the substrate for the biosynthesis of nitric oxide, which is an important modulator for the immunohomeostasis of the mucosa. And of course, threonine contributes to the integrity of the gut barrier, most notably as a constituent for mucin. Threonine is also an important structural component for immunoglobulin, thus contributing to lymphocyte proliferation and activation. So what were we able to conclude from our study? The first is that Eimeria-challenged does indeed negatively impact amino acid digestibility. The second is that most amino acids were linearly affected by the oocyst concentration of a cervulina. The third is that from our meta-analysis, we were able to create our ranking, which showed that cysteine was the most affected amino acid by challenge, followed by arginine, methionine, alanine, tyrosine, and threonine, suggesting that these amino acids may be potentially limiting for broilers subjected to an Eimeria infection. So a nutritional intervention of these amino acids during infections may be key in minimizing the negative effects associated with an Eimeria challenge. Thank you for listening to my presentation, and I'll take questions.
All right, thanks very much, Emily. Nice presentation.
Thank you.
Really interesting results. So when a broiler, for example, has an immune challenge, a gut challenge, one of the things that happens often is an anorexic response. So feed intake is reduced, for example. Now, in the context of your results, you showed decreased amino acid digestibility. So now we potentially have two different challenges from the level of amino acid supply. The third thing that happens is during an acute phase response, muscle protein is broken down to supply amino acids. So did you look at blood levels of these amino acids? Because maybe some of the deficient or some of the impaired absorption of amino acids is offset by an increase in blood tryptophan or blood methionine or cysteine coming from muscle breakdown.
That's actually like a really good point to consider. And unfortunately, with the limited amount of studies that we could find that fit our criteria for this meta-analysis, there really wasn't very many that did report feed intake with blood plasma levels of amino acids. But
Yeah.
I actually am running a second meta-analysis, and I'm going to be focusing more on growth performance.
Okay.
So it should be interesting to see. Yeah.
Yeah. No, it's an interesting area of study. So you mentioned that it might be possible to supplement diets with those amino acids to overcome that. What if that's a protective mechanism of the animal? So, for example, if we look at trace minerals, often during an immune response, the body pulls certain trace minerals out of circulation so that invading pathogens are not able to access those nutrients. Do you think something similar might be happening here?
Oh, yeah. It's definitely a good point. Interesting you say that, actually, because there was another experiment that I ran where I was looking at different levels of, or supplementing different diets with different ingredients as just the sole source of protein.
Mhm.
And you could see that because these diets were wildly varied in amino acid ratios, you could see that there was a difference in the lesion scores, for example,
Mhm.
depending on the diet, really, and the severity of those lesions. So, yeah, it's an aspect to consider.
Yeah. So you probably mentioned this, but were you able to find a lot of studies that fit this criteria, or is this something that?
Not very many. There were more studies that we had included initially, but unfortunately, we just had to take them out at the end because they really didn't fit with what we were looking for. So
Mhm.
it's an area that is up and coming, I would say, and definitely needs a little bit more work.
No, it's great. It's interesting stuff. So where do you see this the recommendations being, maybe in 5 years? So as more and more understanding it, can you predict, or do you have any hypotheses about where you might be in this field?
I'm not really sure, but it's I think as we're moving towards more antibiotic free, that sort of feeding program, there's going to be a lot more interest in this. And so there's obviously, we haven't found any solutions by any means, but as you were saying before, there's no one silver bullet. There's only multiple little BBs that we can contribute. So I think this is going to be something.
Yeah,
Yeah.
yeah, and the more we learn about those nutrition and disease interactions, the better able we'll probably be able to come up with better solutions.
Mm-hmm.
Yeah. All right. Well, really nice work.